Good day, everybody. My name is Samuel Adroaldo, and I work with the Department of Animal Breeding and Genetics, Federal University of Agriculture, Abeokuta, Ogun State in Nigeria. And my area of interest is poultry welfare and insect welfare. You are all welcome to Animal Welfare Group Nigeria with the acronym AWGN. AWGN was founded in the year 2019 after a gathering of students and lecturers at the Federal University of Agriculture, Abeokuta, Ogun State in Nigeria. And this group has three missions. The first mission is to increase awareness about animal welfare and behavior in Nigeria, in Africa, and the world at large. The second mission is to foster collaboration and networking among researchers in the field of animal welfare and behavior. And the third is to educate the public about the importance of animal welfare and behavior. And we meet every fourth and third Wednesday of every month to discuss issues that are related to animal welfare and behavior. And people from different parts of the world are presented on issues that are related to ethical practices, slaughtering, fear, stress, cognition, neuroscience, archery practices, pain, welfare assessment, one welfare, enrichment, vocalization in animals. And when I say animal, I'm talking about livestock, pets, wildlife, insects, and fish. And today's presentation is the 16th in the virtual inaugural lecture series. And today's presenter is Professor Zanella from University of São Paulo, Brazil. And Professor Zanella will be talking about the importance of the early environment on animal welfare. So today I'm the host and I'm also the moderator. And before Professor Zanella will start his presentation, I'm going to read his bio so that people will know more about him. Professor Zanella heads the Animal Welfare Group at the University of São Paulo, Brazil. His trajectory started with a PhD at Cambridge Vet School, continuing in Munich, at the Ludwig University as a postdoctoral fellow. He was a faculty at Michigan State University, USA, Norwegian School of Veterinary Science, and Scotland Royal College. In his current work at the University of São Paulo, Professor Zanella developed research on markers of resiliency in different species, focusing on early environmental factors that improve the capacity of individuals to have a good life. Professor Zanella coordinated the European Union-funded project, Animal Welfare Indicator Project, developing protocols to assess animal welfare, an area that he still conducts research. His team also work on educational approaches to improve the understanding of animal welfare and sustainability among its peers, stakeholders, and interested parties. So, Professor Zanella, over to you to lecture us, talk to us about the importance of early environments in animal welfare. Thank you.
Good morning, everybody. It's a real honor and privilege to be invited for this meeting. Thank you, Samuel, for hosting. And I am going to be, and Olusen was the person who initially invited me, and I'm delighted to be part of this event. Initially, I'd like to indicate that I will be presenting work carried out for my team here in Brazil. And I'm speaking not from São Paulo, but I'm the state of Alagoas in the northeast of Brazil, where I've been participating in discussions about donkeys recently. And very nice. And what I like also to bring to your attention is that I'll be focusing my presentation on the work that we developed using pig as a model, but we have we also have a similar type of approach in sheep, goats, horses, and cattle. Then now, if you allow me, I will be sharing my screen and get started. Okay, I hope you can see my screen now. Then I start then by introducing the university campus in São Paulo, where I'm currently working. Then I had the Center for Comparative Studies on Health, Sustainability, and Welfare. And this campus, this group is in the School of Veterinary Animal Science of the University of São Paulo. And we are situated on a 2,400 hectares campus in the town called Pirassununga. What is really interesting about this campus is the abundant wildlife, and you are all welcome to come and visit us. We have toucans, macaws, capybaras, and eaters, jaguar, and in our group, can you hear me well? Is my sound okay? Is okay? Can you hear me well? Because I'm not able to see you anymore. Can you hear me well?
Yes, we can hear you.
Perfect. Then, when I came to Brazil 12 years ago, and I still kind of work towards that goal, is to create a network of excellence for sustainable development, having animal welfare, better said, one welfare, as a foundation for the sustainability framework. And that's some pictures from our campus. These are toucans. These are kind of deer, marsupial.
So Zanella, is it possible you share your slide because it's not moving?
Oh.
On share and share again. Okay, we have you now. It's working fine now.
No, okay. It's okay now. Can you see? It's changing now?
Yes. Yes.
What is strange that I am not able to do share the screen, but let's see if I.
But it's fine if you are not able to do that. Once we can see what you are presenting, it's fine.
Okay. I want to do this.
I think it's fine like this.
Okay. Okay, good. Well then, and this is the campus, and we're all very interested in seeking international partnership, and especially, and I respond to this invitation with enthusiasm, because in my opinion, South America does very little with Africa, and especially Brazil. Brazil, we have tremendous cultural connections with Africa, meaning there are plenty of opportunities for cooperation. This is just a tree called Jequitiba. A 900-year tree where we organize a real retreat in my group. I'm also telling you that I've coordinated this project called Animal Welfare Indicators, where with 11 institutions in 9 countries, we developed welfare indicators for sheep, goats, turkeys, horses, and donkeys in a EU-funded project. And I encourage you to access the hub. The Animal Welfare Science Hub is a global platform created in the EU-funded project, and the goal is to keep communication going among stakeholders, interested parties. And I totally encourage you to have access to the hub and contributed to the development of the hub. The hub was truly the most important legacy of the AWGN project. And just getting started in my talk, then in my group we have several areas of research. I put in red what I'm going to be speaking today. Then this is the truly fundamental science that my group does, looking in terms of germ cells, fetal neonatal programming, and looking in terms of welfare outcomes. And I'm going to be exploring in details some of the experiments looking at the pig as a model. And just remember what I said. We have similar approach to sheep, goats, and horses and dairy cattle. In my group, we also look in terms of housing systems and welfare outcomes. We work on, in terms of developing automated intelligent systems to assess welfare. We're especially concerned with lameness in cows and in sows. And I'm going to speak briefly on some of the issues that we're interested, that's looking in terms of mitigation of accidents involving animals. We have a tremendous problem with traffic accidents involving animals, both domestic and also wild animals. The domestic as a result of abandonment, and the wild animals as a result of poor planning of roads. Okay? And if something goes wrong, or if you cannot hear me, just go ahead and get in touch. And I think probably Samuel may have my WhatsApp if I needed to be contacted on an emergency issue. I also then in relation to the accidents, we've created this is called the Platform Unified to Respond to Accidents Involving Animal, and this is the logo of this initiative. And in this initiative, we've been creating solutions to mitigate the tremendous problem that is the animals to be hit by cars on highways, or even issues related to transportation of live animals. And this is I've supervised 2 PhDs and 1 master's degree, and all of these candidates were from the police force. And I also supervised a PhD looking at intelligence system to monitor roads, and also I studies from a animal science student, where we studied different ways of putting grass along highways to reduce traffic accidents. This is also another important project in our group. Is called the Zero Abandonment Project. It's a project looking at the roots for the abandonment. And here you see, you see, wait a second. Here you see a donkey, a dog, and a cat. Then abandonment of animals is a really serious issue in Brazil. Then dogs and cats, we've been looking in terms of why are they abandoned, and ways of stopping that. And donkeys, I'm currently at the Northeast, and I have 3 slides in the end of my presentation. They've been also victim of abandonment because of changes in agricultural practices. And this is also incurred in issues related to animal welfare. My program also has an activity under this big umbrella that is the One Welfare. Then we have an extension project that's called One Planet, One Health, One Welfare. And you see in these pictures that I hope you see the on the good side. These are activities on the public square in the town of Pirassununga, where we are speaking on biodiversity, duty of care, sustainability, vegetable gardens, One Welfare, and the relationship between university and educational systems. And this gives you a broad idea of the type of activities that we are involved in addition to the things that I'm going to be talking about. And I go back to my slide, where I'll be speaking then in terms of the early environment and with the focus on pigs. And again, I may refer to some of the work on other species. It became appropriate. And our working hypothesis under this umbrella of the fundamental science in terms of getting a good start in life is that there's an important interaction between microbiota and these cells produced by the immune system in addition to glucocorticoids in the prenatal environment. And this combination of these markers in the prenatal environment, it shapes, alters, modulates the way that individuals may cope, may adjust to situations in life. And I hope that by the end of my presentation, I'm able to persuade you that good welfare, represented by things like environmental enrichment and appropriate housing for animals and appropriate management, good human relationship, can mitigate the negative consequences of this interaction between microbial cytokines and their impact on the resilience and on the offspring. And I did select to focus on the pig model because this is the model that I understand the best. And in fact, the pig model, I can share with you later a link to the special National Geographic, where in 2006, we've been there with my research when I was in Norway, showing that weaning pigs prematurely, you create a memory problem. Pigs become limited in their ability to recognize other pigs, and they fight because of that. That was the start of this interest on the environment, and that started back when I was in the United States. I'm going to be building on a few different ideas. I'm going to speak a little bit about males in the very end of my presentation. I hope the pitch that you're probably going to see my screen kind of small. Are you sure that you can see my screen well? And I'm going to be talking about acute stress that's related to lameness. And I'm going to be looking the outcome in the offspring, looking at the behavior, aggression, and emotionality. Then I'm going to be looking at the effects on the females, in the males, and how that affects the offspring. Can you still see my presentation well? Can you hear me well?
Yes, we can hear you, Professor Zanella.
And the slides, can you see well? Because see if when I try, see, and now let's see, I put now in presentation mode. It has not changed, or?
It has not.
That's so strange. Okay, but then I go back. Good. Then the first piece of evidence that I'm going to be sharing with you is the PhD work carried out by Marisol Parada Sarmiento. She, I supervise her PhD. Marisol now is a postdoctoral fellow at the University of Pennsylvania. And I'm going to briefly explain what was her work about. That was a publication in Scientific Reports. And Marisol was the very first person in the world to report that it matters for a young piglet to be in the uterus of a sow that's lame, meaning that a sow that has chronic pain. And then, then the reason why we thought about lameness as a source of concern is because it is present across different species. In pigs, we know that more than half of sows in commercial systems are lame, meaning walk with difficulties. Cows, over 30% of cows are lame across the globe, probably 35%. Okay. Sheep are lame, maybe 17%, maybe a bit less. And what we know is that an animal that is lame has its social behavior changed, changes feeding behavior, changes physiology, and we understand that changes its mental state. And altogether, combined, there is an impact on the female that is lame. But the question is, what are the consequences for the fetus that are in the uterus of this animal that is experiencing this pain? That was a question in Marisol's work. And the thought process was that an animal that is lame, and again, I'm speaking about sows. We have similar evidence in cows. in cows, calves born from lame cows do go through very similar trajectory that I'm speaking with you now about sows. A lame sow, we thought that may affect their offspring when they're in the uterus, and we suspect the implications are the cytokines, at least for the immune system, and also cortisol. Of course, the placenta has the possibility to prevent the entrance of glucocorticoids through an enzymatic type of activity, but there's a limit to that. And all of these we suspect that has epigenetic mechanisms altering the trajectory of the fetus. And this is in the same area, we have now 3 publications, and that's the very first one. The data set was from a concomitant study looking at levels of fiber. There are kind of 6 assessment of locomotion and also a measure of placental glucocorticoid in these sows. Then we had sows that showed no lameness, 7 animals with 52 piglets, sows that were some moderate lame with 66 piglets, and sows which were severely lame with 38 piglets. In this study, Marisol and the team, they measured aggression by counting in 6 photograph lesions. If you see here, count 1, 2, 3, 4, 5 lesions in the skin of the piglets. And this counting was done by people that were unaware of the state of the sows, if the sows were lame or not. And also she measured fear in these open field tests, where fearful animals would go around the open field and less fearful animals would cross right in the middle. And that's a novel object that also part of the experiment. These are the kind of statistical analysis, but what I'd like to bring to your attention is that for these pigs, if you look at the skin lesions there's an impact on piglet weight, meaning the offspring of the sows that were with the highest degree of lameness at lowest weight on the weaning, and they showed highest level of lesions, especially the animals on the intermediary lameness. Because I just want to bring to your attention that when the sows show high levels of lameness, we end up treating them for ethical reasons, meaning that we discovered that by being in the uterus of a lame sow, the young piglets during the weaning fought more, then they're more aggressive. That's absolutely fascinating. Then we concluded in that first study that for the very first time, there's no literature prior to that relating to this, that pain and inflammation associated with lameness experienced by sows in the last third of their pregnancy altered social behavior. And also we had some data that it changed the fear responses and nociceptive threshold. This is part of a other study published where we identified that the offspring of sows that were lame had different responses to nociception, meaning some very, very fundamental mechanism important for the life trajectory of the individual were altered because of the piglet was inside of the uterus of an animal in pain. And remember, 50% of sows in commercial herd may be lame, and over 35% of cows. And that's a real important issue, meaning the animal is lame. Of course, the lameness affects the animal, affects production in terms of milk, in terms of cows. but also now we know that has an impact, has a really important impact on how the individual organizes its brain and its social behavior. In response to this work, we want to ask the question of what happened in other systems if an animal is lame. Then the studies that I'm going to show you now is when we've used a lipopolysaccharide that is kind of it is a substance that's present in the in gram-negative bacteria, and that produces absolutely these mechanisms probably present in different. We have now the evidence for pigs and for cows, and then we felt in my team we felt that given the high incidence of lameness, we did not want to create lame animals. But the fact that lameness is a multifactorial type of condition, it creates some truly a lot of difficulties. Then what we did, we decided to inject this lipopolysaccharide that is present in the membrane of gram-negative bacteria that creates this inflammatory process that could be, in a sense, equating to lameness. And what we did, we shift a little bit, and then at that point in time, we are concerned a bit on the use of antimicrobials. Sorry, that is incredibly high in pig production, and we decided to monitor change like in vaginal microbiota. And why vaginal microbiota? Because the neonatal, the young pig or the young cow or the young kid, it doesn't really matter. When it goes through the vagina, gets the mother microbiota that populates the digestive system and creates this very rich environment where nutrients are broken down in ways that feeds the brain. Then the vaginal microbiota is absolutely central to getting a good start in life. And then I'm going to be telling you the story on a short format, and I will be delighted to come back to explore the issue further because I know that we have tremendous interest. And this is the work by Luana. Luana did their master's with me, and that was published, and I can give access to the full publication. Can you still hear me? Hello?
I can hear you. We are following you.
Is it okay?
Yes, it is. We can see everything and we can hear you.
Very good. And I'm not sure somehow if people want to stop, because as I change the experiment, if people feel like there's an urgent question, I have no problem being interrupted. I have no problem. If people feel like, or if you prefer me to answer at the very end, you are in control. Just let me know.
It's okay. Thank you.
Then in this study, we started with 42 goats, and we put these animals first. All of them are kept in a paddock like this one here. Then we synchronized estrus. We gave a synthetic form of progesterone to keep them all cycling in a very, very, very similar time format. And then, let's say a week before estrus, we moved them into a group situation, put them in crates. That is it's common in several countries. In Brazil, we were in this period of transition. Europe, the UK moved away from the system in 1999, and Europe moved away in 2013. And some animals kept in groups, 14 in each one of the system. Okay, 6 animals in this system here, 6 in that, 6 that, receive this compound, this LPS, intravenous lipopolysaccharide, that creates like an environment where there is a disease. You create an inflammatory process, activate the release of cytokines, increase the temperature. That kind of lasts maybe for 4 or 5 hours. Okay, just to see how the environment where the animals are kept respond. But our goal is to really focus on the microbiota in the vaginal tract, because that's where, remember, that's where the fetus go through, and that's where the fetus get information about the environment. And I explained that to you. Then we kept the animal in these 3 systems, and we injected 6 of these LPS and 8 as a control. As a control, we just inject with a saline. Yeah, well, just tell that's when the animals got into the system, and that gives you the trajectory. I just want to jump on that. What I did before putting the animals into the system, remember when they are all kept in this, and immediately when we set up them, either the LPS or the saline, we did wash with saline the vaginal tract and recover, and we extract DNA to measure the 16S RNA to understand what is the diversity of the microbiota. Understanding that a diverse microbiota means a more protected vaginal tract. Okay, more diverse, more protected. And then we contrast animals challenged with LPS and saline under the same systems. LPS and saline, LPS, saline. Remember, we had 6 and 8 in each system. And then we sequenced these 36 samples of this vaginal flushing the day before challenge and after the challenge, the 10th day. Okay? Remember, here and there, before giving injection, and then 5 days before the injection. Look how fascinating the results are. When the cells were kept in crates, I'm not sure how it appears to you, like this orange light, the vaginal microbiome diversity was significantly lower when in contrast with the group and also with outdoors, but being significant truly with the group. Meaning, once you inject LPS and a cell is kept outdoors or in group, it doesn't matter. The change in temperature caused by this injection of the LPS, there's no effect on the microbiota in the vagina. However, if the cell is kept on an environment with poor welfare, meaning crates where the animal cannot move, it kind of reduces the microbiota, and especially then the differences in these relatively abundance were found then when contrasted cells kept in crate, in group, and these are some of the phylum that were affected. Bacteroida, cutis, and Proteobacteria were significantly different for cells kept in crates in group. Okay, again changing, but what is fascinating is for the animals kept outdoors, no phyla were found to be significant different for the outdoors kept cells after RPS, meaning that when cells are kept in this high welfare outside, even if they're challenged with this substance, they got increase in temperature. In fact, they recovered quicker than the others, but there's no difference. Then good welfare somehow protect the cells from this challenge. Okay? what did we learn from this experiment looking at the vaginal microbiota in LPS? First, 5 of the 14 gilts kept in crates show clinical signs of urinary infection. What is a problem that it does occur in the system? And cells kept outdoors had quick recovery from the LPS challenge. And in this study, our conclusion was that cells kept in crates, representing poor welfare condition, had the lowest diversity microbiota. In cells kept outdoors, good welfare condition did not alter vaginal microbiota in response to LPS. I brought a kind of a comment here in relation to what Europe did in 2013 and what the UK did in 1999, banning gestation creating for cells. Just remember, this Council Directive 208 allowed cells to be kept in crates up to 4 weeks post-mating. It's too much time, too much time. So in our case, we saw the changes 5 days after the system, meaning that I think we have a problem with cells kept even for the 4 weeks post-mating in crates. Okay. Now I'm jumping to 2 other studies, and I'm going to be speaking on the work done by Patrícia Tatimoto, PhD of Thiago Bernardino, and Masters and PhD of Leandro Sabei. And I'm going to be explaining to you a little bit what were the questions. Then the first study is the work by Patrícia. And the question here is a different one. Remember, I show you that lameness was problem, and I show you that housing in outdoor, that's a good thing, improves the resilience of the cell. But the question that we had in Patrícia's PhD was, what was the impact of, at the end of the pregnancy, if you improve the life of the cell? And we did that by giving cells hay in the last weeks of pregnancy for them to manipulate, to lie down, and then monitor the impact, the behavioral impact on the offspring. And we got the brain of the piglets and look at the epigenetic markers. We use a technique called GBS-MET, and we map methylation profile in 3 brain areas. And I'm going to be showing very, very, very rapidly the impact. And also looked at the repetitive behavior stereotypics, but I'm not going to be talking about stereotypics at this point. Now, there was a study published 2 years ago, and then we had these are cells, identical type of pens. The only difference, we have hay, right? Some cells show these behaviors and reduce. And once piglets were born, we characterize their behavior. And piglets born from cells kept in this environment show less fear. Okay. And then we collected the brain of these piglets, and we looked at the area called amygdala. Amygdala is the center of fear. It's the area in the brain that controls fear response and also has control social behavior, empathy. The frontal cortex is the area in the brain that controls executive behavior and impulse. It's really important for animals and humans that how they organize their days, how they control impulsive behavior, aggression, and also addiction. And the hippocampus is the main hub for emotional responses and memory. And we collected, we extract DNA, and we run DNA methylation using a technique called GBS-MET. Then these are how you test. This is an open field test. This is a piglet. This is a novel object. Then a bold piglet would approach here, come to the middle. A fearful pig would go around here, avoid crossing in the middle. What we identify changes, important changes in the parts of the brain related to emotion, learning, memory, amygdala, hippocampus, and prefrontal cortex as a result of adding enrichment to the end of the pregnancy of the sow. The results show that the hippocampus, remember the area of the brain that controls emotional responses, memory. In humans, the Alzheimer's disease is characterized by compromises in the hippocampus. We understand that some of the changes were affected by that. And when animals show this type of behavior, like they're having a chewing gum in their mouth, repetitive behaviors, in their offspring, we saw some impact, especially in the amygdala. There's some difference in the amygdala. And what we identified that the maternal environment, meaning the presence of the environment, the environmental enrichment, impacted a specific pathway called the neural crest in the frontal cortex. Remember, frontal cortex associated with the executive function, impulsivity, could be aggression, addiction, okay? And this neural crest is an important signal for brain development. And of course, everybody keeps asking the question, you think that's applicable to humans? Probably it's applicable to every species, meaning that having a peaceful pregnancy is probably very good for the brain of the offspring in any kind of species. And both the maternal and this environment and also these stereotypes, they impacted on pathways related to this ethanol metabolism and lipid signaling in the hippocampus. Then the hippocampus was affected in both ways, either by this or the case of cells showing this behavior, the mother of the young pigments. And the amygdala, remember the amygdala is the center of fear. And also there's been recent evidences linking amygdala to autism and also empathy, social behavior. And what we identified that this maternal environment, meaning having a rich environment before parturition. This is a pregnant animal with piglets in their belly. Had some changes in these epigenetic markers in pathways associated with microtubule polydepolymerization. These are incredibly important for the offspring to form memory. And this, we quoted the study. This is our own study where we did in sheep with Marjorie colon, whereas I was in Norway. Then in conclusion, environmental enrichment in late pregnant cells altered epigenetic markers in the amygdala, hippocampus, and frontal cortex of their offspring, meaning that taking care of the pregnant animal, it doesn't matter if it's a cow, if it's a sheep, if it's a goat, or it's a sow, has positive implication for the offspring. Okay, now I've done with the second part. What I'm going to be doing now is moving to males, because is everybody following my talk? Is it okay? Am I speaking too fast? How are things going?
Fine. People will ask you questions at the end of your presentation. I've listed like 5 questions for you, and I know our
Very
viewers
good.
are also doing that.
What I'm going now to do is to focus on the study on boards. This, I show you the picture of these 3 gentlemen here. That this is Patricia just spoke about the study now. Thiago and Leandro. Thiago focused more on the impact of the housing on the boards, and I'm not going to be talking about. And Leandro focused on the impact of the housing of the boards on the offspring. And this is probably one of the most fascinating research that we've done in the recent years. We asked the questions if it matter how the boars are kept, the father of the offspring. And that's Leandro. Leandro now is a postdoctoral fellow at the University of Pennsylvania in the United States. Then we'd like to ask if housing boars in different ways had impact on the offspring. Remember that but plenty of evidence that affects sows. And then what Thiago and Nando did, they house boars in crates, and housing board in crates common in stations that collect semen, okay, in pens. Or what happens if you create an environment where boars receive the enrichment, meaning they get showered, they have hay like the same like the sows got, and they get groomed with a broom twice a day, like they are in a spa. Okay, pen, crates, and spa. Very good welfare, good welfare, good welfare. Okay, and then I'm going to repeat just to make sure, because this the methodology is a bit complex, and I'm going to be repeating. I remember we kept boars in 3 systems. For these are boards kept in pen, pen with enrichment in crates, and we kept. Let's go to the next. The next one is. Then yeah, these are how the yields are kept. But the central to what we're talking about is we kept then the animals in 3 systems. Okay, so the animals are kept either in system with enrichment, in pens, or in crates. And what is exciting, I'm going to tell you about, we mix the semen and had a pool of semen. I just explain a bit in a bit better way. And that's how we kept the sows. That's where how the piglets were kept during maternity. That's how we test emotionality. Then the youths are kept there. The piglets were born and housed there. We test emotionality in this open field where we release the piglet. I had a small move that ended up removing, and the piglet either moved here or across in the middle. And we test in this open elevate plus maze, meaning that we release the piglet here. A bold pig would walk in these open arms of the maze, and a fearful pig would cross in these closed arms. Remember, this is elevated, it's a maze, open arms, closed arm. And then we had I'm going to be showing some data when we pigs at 29 days. And I'm going to go through again the methodology, but again, we measured skin lesion, people that were unaware of the treatments in these piglets, and count the skin lesions on 6 body photographs during 5 consecutive days to measure progression. And just remember, and then all of these tests that we've done, these tests here, these assessment here, measuring of the skin lesion, and even nociception using this algometer where we press these different areas to see how much pressure was needed for the animals to have a behavioral response with this von Frey analogisometer. Okay, and then when we finish all of these studies, remember we had no idea which father was. We sent paternity tests to be analyzed. Just to understand, then we had the boars kept in crates, in group where we enriched, we groomed this animal twice a day, we give hay, we give shower. We mix the semen, have a pool of semen, but we characterize the quality of semen beforehand. But then we mix. Then we had a pool of semen of high, medium, low quality, and we inseminated 5 youths with a pool of semen, meaning we had no idea who the father was. And then when we finished everything, we did the paternity test. Okay. Then all of these behavioral tests that I told you, the elevate plus maze, the open field novel object, the nociception, whatever we did, we had no idea if the piglet was son of a boar kept in crate, in pen, or in enriched pen. And we mixed the semen right before insemination the sow. Then we had no idea. See, again, remember, we and I say several times just for you to understand how robust was. Then we classify the pigs as high, medium, and low in terms of semen quality. We had animals in crates, in pens, and enriched pens, and we prepared 6 pools: a pool of semen from the high quality semen, a pool of medium quality semen, and a pool of low quality semen. And this pool we inseminated yields. We mixed right before we inseminated. And we did that just to make sure that the maternal side was not a factor. Okay? And then we end up with 13 because we had 5, 4, and 4. Remember? We got the animals for 40 days in the system, either in crates, in pens, or with enrichment. 40 days later, we collect semen. We classified the animals either had high fertility, medium, and low fertility. We got 2 from crates, 2 from pens, and 2 from enriched from each one of these. Prepare a pool of semen, inseminate the ewes. Prepare a pool of semen, inseminate, prepare, inseminate. Then, and we only knew who the father was when we finished all data collection. We finished all data collection, then we refer back to who the father was. Okay, and then we these are the pigs that we in, and the whole. I just can go back here. And there was an expression of a boar being groomed with this broom twice a day. We have now 4 publications of these studies. This is the first one, showing that the way that the boars are kept affect survival of the offspring, aggression, and nociception. Quite fascinating, this article, meaning that when piglets were, when pigs are, more piglets are born. Remember, we mix the semen just before insemination. We found that more piglets are born when their fathers were kept on an enriched environment. More pigs are born alive, and more were win, meaning that there are higher mortality when the piglets are kept. They're kept in crates. Then crates not good for development boars. Surprising, we had a trend that's P053. There's a trend in the number of skin lesions on the left side. Remember, count the skin lesions. What we found out is that animals kept in crates appear to have high number of lesions, contrast with the 2 other systems. In a second publication, Leandro explored the emotional responses of the offspring. Remember, the father were kept either in crates, pens, or enriched. And the question was, how did they respond to tests of fear and anxiety? And I showed you this, the elevated plasmase. Remember, these are the closed arms, kind of a fearful pigs would go here. These are the open arms, kind of less fearful pigs will go there. What Leandro found, there was a difference between crates and enriched. He used a multifactorial statistics to combine the impact of that. But the factor that he called activity fear shows a difference in the offspring when the boars, the father of the offspring, were kept in crates, contrasted with the enriched pen. And in similarity, also when contrast crates and pens, there's a difference in exploratory behavior. What I'm trying here, I'm not going to get into the detail. What is the meaning of that? What I'm telling you is that having a father, a boar, kept in crate was sufficient to alter the trajectory of the offspring. That's absolutely fascinating. And I conclude by saying that the studies that I should share, they're building evidences first to understand this puzzle, to explain the interaction between housing, microbiota inflammation, pain, and welfare, and the models that represent, especially social behavior like aggression in all of these studies, and nociceptive threshold are incredibly important for coping strategies are disrupted, creating a less resilient offspring. I finished my presentation on the data. I had now a few slides mentioning what I see the common potential areas for us to collaborate with Nigeria. Then I made a few. Then the first one, are you still with me there? Can you hear me well?
Yeah, very well.
Okay. Then what I feel is that Brazil and Nigeria will share similar things in terms of the importance of producing food. In Brazil, we still have a part of the population that lives below poverty line with difficulty in accessing food. Things are changing in a positive way, but it's still a problem. And there is a need to produce health diets at affordable price for all in a sustainable way. We are experiencing also massive risks to the planet's biodiversity, and it's often being present risks to animal welfare and concerns in relation to human welfare. And of course, the climate crisis is a growing threat. Brazil is going to be hosting the COP30 in Pará in November. And of course, these are kind of it's not only a matter of lacking of food, but inequality in excess of food, poor use. And this is the Nobel Prize. There was a Brazilian preceded the people at the FAO. This Hunger Zero that was launched by a Brazilian who was no longer there when the FAO was awarded the Nobel Prize in 2020 for the Hunger Zero. Of course, the climate change issues are massive, and we need to be thinking about animal welfare in this kind of context in terms of biodiversity. And what I feel is that Brazil and Nigeria, with this abundant wildlife and rich biodiversity, have a role in I mean African countries. I mean the African countries. I mentioned Nigeria, but I mean across African countries, we encounter an impressive biodiversity, and this biodiversity is absolutely under threat. And we, I had the possibility to collaborate with some colleagues in the past in Botswana, in Kenya, in Nigeria. These are the few countries, but what I feel is that we need to know more. I mean, what I propose is a summit between African countries and probably Latin American countries, trying to identify the common threat under the sustainability framework. I put here the photo of Priscilla Komolaf. We were collaborating on a project looking at slaughtering practice and health status in the Ekiti abattoir in Nigeria. We've been working together since our encounter at the meeting at the ASA in Curitiba last year, and Priscilla has been collaborating for almost a year, trying to understand the issue of animal slaughter. And again, the issues of slaughtering animals, it is a problem in some areas in Brazil in terms of animal welfare. There are commonalities to be resolved there. I told you when I first started my talk, the international commerce of donkey skin is a huge issue. The skin is for the production of Adroaldo, this traditional Chinese medicine. And in Brazil, the animals are captured, especially in the northeast of Brazil. I've been involved in a task force in the past. Just to give an idea, these are kind of places where people bring donkeys, and donkeys, they're truly important in the northeast in terms of their resilience, help small farmers, and is a kind of a cultural type of symbolic item. And just to give an idea, that was the population of donkeys in the 60s. This is our own publication that we published over in 2021. That was less than 400,000, and over this period, 240,000 donkeys were slaughtered between this period. really, really massive challenge in terms of population. These are donkeys, and here are mules, that is hybrid between donkeys and horses. Massive, massive challenge. And I mentioned in the beginning this idea of the Center of Excellence. So we build capacity in our group to create, to learn how to use multimedia to communicate with society. And I answered questions by Samuel right at the beginning, what I consider to be the most important challenge in animal welfare, and in my opinion, is our modest ability to communicate with civil society, because there is a general understanding of the importance with our relationship with other animals, but our communication with society, and it's really poor. We need to learn. We need to get together to devise better strategies to communicate with society. And this is Marcia, who basically handles that of communication in our group. And then my approach to research, I hope that you observe that the different expertise of the collaborators that I engaged in the publication, because one of the characteristics in my program, I collaborate intensively, and I learn a lot from people, from expertise and credentials that I do not have. And I do encourage people in animal welfare to do the same. Be close to people who work in animal nutrition, animal genetics, animal health, human-animal interaction. You need to build network of cooperation. I'm absolutely committed to high-quality research, and I love to collaborate with multidisciplinary teams. In my team, we are all inspired by novel ideas. We always have time to discuss things over coffee, walks, retreats, and meals. And I have always undergraduate students running very complex experiments together with master's, PhD, and postdoctoral fellows. I attract very young people in my team. And in this talk, I've only shared with you data published since 2021. And I do have similar credentials to other programs and other species. But I do understand that focusing on this latest research gives you an idea of the focus that we are conducting. Then I finally saying that we are going through what I call is a perfect storm. Animal welfare, human welfare, and sustainability issues are better addressed under the concept of one welfare. And I think there's a massive opportunities to improve human welfare operating under this framework. I would like to acknowledge people that are helping with that. These people help me discussing some of the things: Marisol, Leandro, Luana, Thiago, Arthur, Marcia, Paulo, all of the people help with that. This is my family, wherever across the world. This is Isadora, Mauricio, and Marcia, and we go across the world together. They are blessed to have. And I also like to acknowledge people that cooperate with us, the different labs. This is logo from our team, corporate lab, my department, other universities, Brapa, funding agents in Brazil. They're quite important, especially FAPESP, CNPq. These are important funding agencies. And thank you very much for being patient with me. And I now finish my presentation.
Zanella, that was a very fascinating lecture, linking animal welfare with microbiome, epigenetics, and animal behavior, and I really enjoyed it. Before people start dropping their questions in the chat box, I have a few questions for you, and I hope you will do justice to those questions. I look at your results for the housing system, looking at the crate, the group house, and the outdoor, the pen, and the crate, and the enriched housing. And I discovered that we have more skin lesions in the crate. Then we have the lowest diversity of microbiota in the crate also. So how do we convince pig farmers, that keeping their sows in cage is not good for pig welfare? You know, we talk about communication. How best can we convince them? I'm asking this question because there was a time I was doing my postdoc in Purdue University in the US, and I was reading news that we have some farmers using crate system for their sows, and the government is saying no, they should not use it, and they took government to the court about this. So how best can we convince them that crate is not good for sows?
This is a really important question. What happened, Samuel, is that we need to be working together as a scientist, because we scientists told farmers to put sows in crates. The system, farmers did not wake up in the morning and say, look, let's put sows in crates. I mean, the process started with the scientists. And you find, what you find is that you see tremendous amount of resistance among our peers. And in my view, what we need to do is to discuss among peers. And truth of the matter, in Brazil, the pressure comes from society. Brazil is changing. We have a new normative that established the end of crates, but puts a very long deadline for things to happen. But society says no. And the transition for farmers, in my view, needs to come with resources for the transition. The transition is very costly for farmers. And farmers in peak production system operate with low or relatively low margin and a lot of oscillation in prices. And the transition in the UK in '99 was very costly. A lot of farmers end up getting out from production. Then, answering your question, in my opinion, you need to build a trustworthy relationship with farmers. People need to know that you're operating on their best. And what we need to do, we need to challenge young people to get new publications with assessment of costs from the system. And truly, the evidence that we have now, and we published from my PhD in Cambridge, together with Mike Mendel. Mike Mendel was a postdoc, was assessing the welfare in housing systems. That the evidences that keeping sows in crate are tremendous. I mean, to go to the data from Jeremy Marsh and Ford showed that they have a lower muscles, lower bone density. There's no evidence showing that is a positive one. I think what we need to do is to set up a transition, and eventually, like Europe did, like the UK did, probably need to be somewhat on a legal framework to get the meaning that. But the legal framework needs to come after creating this relationship and talk to farmers, showing the data, and stop. And I respond because in Brazil happens the same, and there's a lot of conflict information getting to farmers. We need to get a proper and clear guidance regarding the negative aspects of crate. And I tell you, I'm convinced that keeping sows in crates is not sustainable. It's not, and for the reasons that we've been showing for many years. But then, just to be precise in the answer of you, first, we need to talk to farmers. No, before. First, we need to talk among peers to get a clear message. Second, to talk to farmers. And third, we need to create mechanisms for the transition. And for new projects, I think the answer should be immediate. You should be able to stop that. But the systems of keeping sows in crate is not sustainable. Not sustainable at all.
Thank you very much. And then Dr. Priscilla is asking here that it's very difficult to keep pigs in their natural environment in Nigeria. So she's asking, how best are we going to achieve this because of the financial crisis and limited resources?
Yeah, if you don't need to have kids outdoors. You have systems kept in groups in indoor pens. Mm-hmm. I cannot speak about the systems in Africa because I only visited South Africa. I've never been in other parts of the African continent, and I'm really keen in getting to know better the reality. But there are systems, I'm sure. And then and when I meant in my presentation that Brazil has tremendous possibility to learn from Africa and to share things with Africa. We have different system of keeping pigs in Brazil. You don't need to put them outdoors. No, not necessarily. You can keep them in group housing. There are different ways of keeping pigs that you do not need to put outdoors. And some of the systems are less expensive than crates. Then with the transition, what I feel is that we operate with too many conflicting information among our peers. I think we need to get the information organized among the peers to come up with decisions like that. But the opportunities for a sustainable pig production system are tremendous.
Thank you for that. I hope Dr. Bisilia is satisfied with that response. I was looking at your experiment on epigenetics, where you used the brain of the pigs. I'm looking at the invasiveness of that procedure. Is there no alternative to that? Because I'm like, when you need to remove the brain, I'm kind of bothered because it's invasive. How did you do that? Or is there
an
alternative?
I'm glad you asked that kind of question. I mean, in my research group, our approach is always an approach of minimizing thermal experiments. But I don't want to sound like I'm contradicting my role in animal welfare. If we make a decision to have a thermal experiment, we do things incredibly carefully, and I feel that putting down an animal, killing an animal, if done in an appropriate way, you may have some ethical issues with that, but there's no welfare issues. And then these piglets, they're killed, they're killed on a protocol where we took care that we mitigate suffering. Of course, that can be, but we made sure that our research group talked through, and that we made the decision based on the potential outcomes of the measure. But experimental work, when we talked about that, and we've done a few. We tend to do less and less of this thermal experiment. But of course, if I had access to magnetic resonance, looking at imaging, we could potentially see that. But at that point in time, we did not. And in fact, I mean, imaging process would give us much better insight of the brain than get to the tissue and looking at the markers. Meaning there are far more elegant techniques. Probably the only problem, we had no access to that at that point in time.
Thank you very much for that thoughtful response. Somebody's asking here if the model you have used in pig is applicable for other research animals.
Sure. I tell you, we have a very similar responses in cows. Calves born from cows that are lame had differences in pain responses. They have also differences in how they have inflammatory response, meaning that the fact that we saw in lame sows, we also identified in the PhD of a veterinarian called Bruno Steiniger, who have the same response, meaning that to be in the uterus of a lame cow is very bad. It's as bad as to be in the uterus of a lame sow. And we have some data in sheep where we had positive handling of sheep and then late end of pregnancy, and we looked at cognition, and if you're positive with sheep, the lambs are better in their cognitive responses. And we also look at the pattern of spine formation in the brain that are the link for the synapses, and we did find that there's an impact in these synapses in lambs that received negative interactive. The negative was just not to be physically aggressive, but just behavioral DSC, uncomfortable for the use. Meaning that, and we also done some work in goats, where by being negative to goats, we alter the responses for the kids in the behavior and also in the placenta. Then we have very strong models for sheep, goats, and cows. And now we're building models on horses right now.
You very much. I was following through your presentation where you classify the semen of the boas into high quality, mean quality, and low quality. If I followed you where you classify based on the housing system, am I correct?
Yeah, we use the CASA system, it's a computerized system. We did that because we wanted to contrast the housing controlled by the quality of the semen, meaning that we use this computerized system called in Portuguese CASA, C-A-S-A. It's a computerized system to analyze semen quality. And then we got the boards that are the top ranking, middle, and low, and we select them on the basis of their housing. Then we just prepare the pool of semen on the basis of this quality of semen, just to prevent it to contrast things that are not comparable.
Thank you so much. Then, in one of the experiments where you use the brush to groom the pigs,
Yep.
how many minutes did that take for each pig?
2 minutes. 2 minutes twice a day. Yeah.
And what was the experience like? What was the response of the pig? Behavioral response?
Amazing. I mean, I had a small movie that was done initially was done by this he's a professional, Thiago Bernardino. Pigs respond to these.
I know Thiago.
Yeah, and it's really massive because pigs are very tactile, and Thiago did a trial. Thiago, in fact, I didn't bring Thiago's data, but Thiago did some work looking at the impact on the semen, and what Thiago found is that housing system affect semen quality, some aspects of semen quality, and some aspects of the testicles, and but the boars they really relaxed. I mean, they show a very clear response, positive response to this grooming. Thiago did that twice a day. There was a combined work by Thiago Bernardino and Savay.
Okay, thank you very much. Somebody is saying thank you for your great presentation. That is from Mayola Oluyemi. And one last question from me, and let's see if other people will ask questions. I was wondering what the role of genetics would be on your first experiment where you challenge with a lipopolysaccharide. You use a breed of pig, and you have your result. If you use another pig of breed, do you think the result will be different?
Yeah, I don't know the answer, but the chances are, yeah. But some of these mechanisms, we use LPS in sheep, and I mean, answer your question is I have no way of being sure about that, but it depends how different are the genetic strains. Then the possibility that the genetic impacts are always there, and but that's why we decide to contrast in the same genetic strain. But there's always a possibility of this interaction between genetic strain and the responses. Yeah, then there is a possibility. I would be surprised. I did not show some of this. We looked at the impact LPS on the corpus luteum, gene expression of the corpus luteum, and the changes are quite strong. But again, we make massive progress in selection, and the chance that the new research needs to be done are quite logical. Thank you for the question. It's a real possibility. I don't think I can give you a clear answer on that now.
Thank you so much, Professor Zanella. Is it possible to reach out for people to reach out to you if they need training?
Absolutely.
Some other things.
Yeah,
I also want to ask if you have some training positions in your lab, maybe PhD or masters, but if people can reach out to you.
You have my email address. I mean, I'd be delighted. I'll be answering questions probably after Monday. I'm now on the middle of a holidays. Then after Monday, I'll be delighted to get messages and positions opened in the lab. At the moment, we do not have any funded positions, but we're always very interested in welcoming new people, and I feel like we need to build this bridge between I would say not only Brazil but Latin America and Africa, especially Brazil. You'd be surprised. You'd be really surprised how common cultural issues we have between Brazil and several African countries. I mean, I tell you, in expressions, in music and dance, and then what I feel is that we should collaborate much more, and the same type of
Okay, that question is from Safian from Ghana. Safian is trying to ask that we know that hard concrete floor affects the leg of pigs. So it's now saying, how does enrich out concrete floor not have negative effects?
Yes, you have different type of concrete. Some people use rubber mats in the floor. You can put bedding. I mean, the reality. I mean, the way that we have there are good concrete and bad concrete, and some bad concrete cause abrasion. They're abrasive to the feet, but there are different ways, and to me, having a very good flooring system is essential. And I think the comment from this gentleman is very precise. We have very bad type of flooring system, and there's a demand for us to carry out more research. What is the best way of catching pigs indoors? And there's some deep bedding system. There's several ways that people are housing pigs indoors in floors that are less abrasive. But the issue of the flooring is quite a serious one and needs to be addressed quite rapidly.
Thank you very much. I hope Safian is satisfied with that response. Do we have any person with questions? Okay, Asot is saying thank you very much. So
it's a real pleasure. It's an honor to be in this group, and I remain available for additional questions, hosting visitors, and if you identified any funding possibility for joint collaboration, we'd be delighted to do that. And I'd like to motivate you to check the diversity of credentials in different areas that Brazil has. And I think we have the opportunities to host people here, and I think Brazilian students would benefit enormously spending time in Africa too.
So thank you very much.
Pleasure.
Thank you, Professor Zanella. And I want to thank everybody for participating in today's presentation. And I hope we enjoyed the lecture where Professor Zanella linked animal welfare with microbiome, housing system, behavior, and epigenetics. And we hope that people will join our subsequent meetings. And a few announcements. The International Society for Applied Ethology Conference is holding in the Netherlands from August 4 to August 8. We want our people to travel to Netherlands and present their work on animal welfare and behavior. I will be there. I will be talking about how handling human-insect relationship affects insect welfare. Then people can join us on our social media. On Twitter, that is X. Our Twitter handle is at AWGN14, at AWGN14. You can also join us on Facebook and LinkedIn. Our LinkedIn and Facebook name is Animal Welfare Group Nigeria. So we see you next time, and we hope everybody enjoyed this. And Prof, thank you very much for that wonderful presentation. Thank you, everybody.